Hi everyone, my name is Philippe Auchamp and I'm currently a PhD student in bioinformatics at the Computational Biology Unit of the Duve Institute, which is part of UC Louvain University based in Brussels in Belgium. My PI is Professor Laurent Gâteau. In this short series of videos, I'm going to introduce and demonstrate the use of a tool called CytoPipeline. CytoPipeline is a suite of R packages that I'm currently developing as part of my PhD, and which is aimed at building and visualizing pre-processing pipelines for flow cytometry data. So we are in the context of analyzing flow cytometry data, and we want to do it in an automated and reproducible way, so using a data analysis pipeline. So such a pipeline is typically split into different parts, from sample acquisition to data pre-processing, then typically cell populations identification, and finally downstream statistics. Here we focus only on the data pre-processing part. What's in there? A series of different steps that we need to apply before analyzing the data. So for example, compensation, in the case of flow cytometry, this is very important. We have scale transformation, then a series of pre-gating steps, so eliminating undesirable events, and so on. So we have to consider that building good pre-processing pipelines in an automated way is quite challenging. Why? Because there are so many available methods for each of these steps that it's very difficult to decide which steps to choose. Also, the order is can vary from one pipeline to the other. So how to assess what is the best pipeline, how to create good pipeline for a specific purpose. That's where CytoPipeline comes into play, because it provides a series of very nice tools to help designing such pipelines. For example, very nice feature is the visualization tools that CytoPipeline provides. For example, here we have the viewing of the different pipeline steps. Now we can also investigate pipeline steps, so digging into it to see and diagnose what happens behind the step. Also, we can compare two different pipelines. So, for example, a pipeline using a specific quality control method against another one and see what is happening. Also, CytoPipeline provides the ability to visualize the scale transformation that has been generated, which is very handy as well. On top of that, CytoPipeline also provides support for pipeline design and run, for example, by centralizing the pipeline definitions using a centralized text file to define the pipeline steps. Also, there is support for controlling the execution, and also other very nice tools like disk cache, support for parallelization, and so on. And that's it. So if this short introduction has raised your interest, you can do two things. One is looking at the GitHub repos of CytoPipeline or my poster, or the second one is to have a look at the rest of the videos of the series. Thanks a lot and see you soon.